Hello, everybody. Good afternoon or good morning to you, depending on where you happen to be in the world today. Welcome to the first IPC Journal Club of 2025. Very excited to have Alex Sundermann with us today to talk about genomic surveillance for infection prevention and control. This is going to be based on actually a really exciting preprint article that Alex has on medRxiv at the moment, but we'll hear all about that in a moment. But before we get to that, let me just give you a little overview of IPC Partners and the journal club that we're trying to run, and we've been doing for maybe 6 months now. So the aim of the journal club is to provide some insight into a topical paper, and none more topical than genomic surveillance in IPC, in my view anyway. We want to try and generate some discussion, so there'll be plenty of time for your questions. When we get towards the end of the session, please do plop your questions into the Q&A box or comments into the chat. We want this forum to provide opportunities for CPD so that we can all develop and learn. It's a good opportunity to exercise critical thinking skills, to look at new things that maybe you haven't looked at before, or maybe look at things you've looked at before but in a slightly different way. We're keen to build confidence for your own independent research and study, ideally to foster an expert network of enthusiasts, and in an ideal world, have a little bit of fun. So the Journal Club recording will be published after the event. And just as we get started, I'll put a little link into the chat. We've got a poll running at the moment to allow you to rank what you see as the barriers to genomic surveillance in an online poll, and then we'll look at that as part of the Q&A. So with all that being said, I'm going to stop sharing my slides and hand over to Alex to talk to us about genomic surveillance for IPC. Over to you, Alex.
Thanks so much, John, and thanks Phil and John both for inviting me to share some of this. I'm really excited about some of the discussion here at the end. But yeah, very excited to share one of our recent preprints. So preprint, means not peer reviewed, but we're currently finding the right journal for it. But it kind of highlights, multiple years of research and trying to implement a genomic surveillance program. So in this, brief few slides here, going to highlight some of that and have hopefully a great discussion. Disclosures, you can see them here. I won't be talking about any products though during this. So very quick about me. John and Phil and I were actually just chatting about some of my background. So I actually started out as an infection preventionist many years ago. I worked as an IP for 7 or 8 years after getting my MPH, and I did a doctorate of public health degree while I was an IP. And then after finishing IP, I joined the dark side of academia, where I do a lot of research on outbreaks now. But I keep the best of both worlds because my research and outbreaks, I get to interface directly. You can, out my window here is the hospital where I worked at and where we do our research. So I get to work with the IP team every single week to try to investigate some of these outbreaks. So I'm loving being able to still work in IP while still doing research here. So very broad overview of whole genome sequencing. If you're not quite familiar, what whole genome sequencing, or sometimes we say genomic sequencing, allows us to do is essentially DNA fingerprinting, where normally you have a positive culture of an infection, and we can't definitively say, even by looking at antimicrobial susceptibility test data, did patient A give it to patient B? What genomic sequencing allows us to do is say, these two infections, these two MRSAs are exactly the same, meaning patient A gave it to patient B, or patient A and B got it from some common source. Now, I am simplifying a topic that can get very complicated and very complex to a point that even, I can't fully explain sometimes as well. But this technology is relatively new. We used to use a technology called pulse gel electrophoresis, PFGE, but only in like the past decade or so has whole genome sequencing been used. So how is genomic sequencing used in hospitals? What we say is kind of, the typical standard approach is this top level right here is what we call reactive whole genome sequencing. And now what that is, typically in a healthcare setting, you have infections that will occur, and we do surveillance, are they hospital acquired or not? But oftentimes it takes some astute clinician to say, I had 5 MRSA infections on my unit this past month. That's weird. Let's do whole genome sequencing on some of those to see, are they related? Oftentimes what we see, what other people see, is that, you have very few, if any of them, that when you sequence doing this approach, they're part of an outbreak. Oftentimes we see none of them are genetically related, means we didn't have an outbreak. That's not to say that they weren't hospital acquired. It means that, they weren't part of the same source or transmitting it to each other. So your interventions might be a bit different. What our program here at Pitt and other people as well have been researching is the concept of genomic surveillance, meaning sequencing almost every infection as they occur. Because hypothetically, if you're sequencing all of them, all it takes is 2 infections of the same to detect an outbreak or a cluster to try to identify the transmission route and make interventions. And hypothetically, you could prevent that outbreak from ever spreading to 10, 20 plus cases where sometimes we see them getting that big. So how do we test this hypothesis? We did it in 2 different phases here at Pitt. The first we published a few years ago in the Journal of Clinical Infectious Diseases, where we did genomic sequencing surveillance for 2 years, but we didn't tell the results of those infections to our infection prevention department until we finished the study period, because we wanted to see what does current infection prevention practice, meaning reactive whole genome sequencing, find versus what is genomic surveillance find. And what we found over the course of 2 years, sequence about 3,000 patient infections. You can see on that figure there on the left, a lot of Pseudomonas aeruginosa, a lot of C. diff. We found 99 outbreaks with 297 patients. And these outbreaks range between 2 patients up to 15 or so. You can see the dark blue there near the top. We published one of these separately, was an outbreak of VRE related to interventional radiology. So this outbreak went undetected. These patients were on multiple units spread out across time. The only thing in common was interventional radiology when genomic surveillance detected this outbreak. And we were able to find that unsterile contrast was essentially being injected to our patients with 1 technician working in interventional radiology among all these patients. So what was interesting is the same time frame, IP requested what they thought were 15 outbreaks of 130 patients. Only 5 of those patients were genetically related, meaning that genomic surveillance detects many, many outbreaks that go undetected, and current methods misidentify multiple outbreaks. We are also able to do some modeling that shows, even with the cost of sequencing all of these patient infections, if we had implemented interventions in real time, we actually could have saved net costs by preventing infections and the treatment costs associated with those infections, because the cost of sequencing an isolate can go as low as about $90 an isolate, but the cost of treating a pneumonia, for example, can be $20,000 or so. So you prevent 1 pneumonia and you can sequence multiple patient infections. Excuse me. So we had some really promising data and we said, okay, let's don't do this in real time anymore. Let's start doing this every single week. So that's what was the premise of our preprint here. So every single week, we would, our clinicians suspected they had an infection. So as routine, they would send them down to the lab twice per week. We would go down to the clinical lab, collect them, bring them to our research lab, sequence them, analyze for outbreaks. And my job would be to review those records for commonalities of what could be causing these outbreaks or how could they be transmitting. From there, what's different now each week, I would work with our infection prevention team, make interventions, track the interventions of what we're doing, when we're doing it, and continue to monitor for more infections downstream. So what did we find over the course of 2 years? So this is the preprint data here. We started in November of 2021. We are still doing real-time weekly genomic surveillance to this day. But the cutoff time frame for this preprint was 2 years. So November of 2021 to November of 2023. We sequenced 4,700 patient infections during that time frame, representing about 4,000 unique patients. We found 172 outbreaks of almost 500 patients. You'll immediately know that the amount of outbreaks is higher, but we're going to touch on that in just a second. These clusters range between 16 patients. Our turnaround time, which we get asked about a lot, so the time when like a culture was taken at the bedside, you take that blood culture, to the time that we have genomic results is 15 days. And in our experience for bacterial outbreaks, that is fast enough because the timeline of a bacterial outbreak we find is quite protracted. Flip side, if you're doing respiratory viruses, we think that needs to be a bit faster, but that's a separate topic. Among our outbreaks and our outbreak patients, we were able to identify possible routes of transmission about 60% of our patients. So what were some of the interesting outbreaks? We had 9 endoscope clusters at 2 patients. I highlight 2 patients because typically at our center and other centers across the world as well, we used to see endoscope outbreaks even go undetected about 10, 20 plus patients until we were able to find them. Here we have 2 patients, the exact same bacteria, the exact same endoscope was used on them. We immediately remove the scope, that outbreak stops. Whereas we used to see the outbreaks grow much, much bigger. Interestingly, too, one of those endoscopes that we removed, although we cultured some of them and they didn't grow, one of them failed a leak test and had all these issues inside, but we weren't able to recover the isolate. We had some outbreaks related to shared ventilators. One ventilator, we typically used to think that ventilators stayed on the same ICU, but we had one patient in one ICU go on a ventilator, get the infection, unfortunately die. That ventilator moved to a different unit and then put on a second patient, and that patient developed the exact same infection. We had multiple clusters from outside facilities. There was one really interesting cluster of 5 cases of Proteus mirabilis. These patients were being admitted to us uroseptic from a ventilator-skilled nursing facility north of our hospital. And after a couple, we kept notifying that facility. You keep sending us these patients that we're culturing and sequencing. They're all coming from your hospital. There's nothing in common here. They took a look at their ventilators and found that the single-use ventilator filters were never being changed among the patients. So they were able to adjust that. We had a national eye drop of extreme drug-resistant Pseudomonas related to contaminated eye drops that we had 2 patients that with the same infection. But at the time, we didn't have any reference genome, meaning a publicly available genome to compare to that national outbreak. So we put it aside. Months go by and the CDC here in the US releases a reference genome, and we found that we have a direct match. When we look into one of the patient's charts a bit closer, we found that they had eye drops from Amazon, the national retailer, right before their chronic infection started from here. And then we also had a pseudo outbreak of contaminated autopsy blood cultures where patients died at various hospitals, came to us for an autopsy, and their blood cultures, although not collected sterilely, had matching Serratia or Pseudomonas in the blood. When we did an investigation, we found that in order to move the patient onto the table, they have to hose down this table. And from that picture there, we cultured the sink and found the matching environmental isolates to our contaminated blood, autopsy isolates as well. So a bunch of interesting outbreaks. But what did we find after we made an intervention? So as soon as we made an intervention, 89 of the outbreaks stopped immediately after 2 cases, meaning we saw no further cases grow in the outbreak once we made an intervention. There were 20 outbreaks that kept on growing, but only on different routes of transmission, which we had not yet intervened upon. And then we only saw 5 outbreaks continue on the same route of transmission after an intervention. But we have some data that I'm not going to necessarily go into today that shows why those interventions might have failed. There were 58 outbreaks with no identified transmission route, but I have some additional data on that at the end. So the big question is, we hypothesized in the past we could have prevented infection. We're doing these interventions now. How many infections did we actually prevent? So we wanted to use that retrospective genomic surveillance data where we made no interventions. But we wanted to track not necessarily how big the outbreaks get, how many outbreaks get, what are we actually doing? We're making an intervention and we're trying to stop on the outbreak on the thing that we're intervening on, the unit that we're intervening on, the piece of equipment that we're intervening on. So what we did, starting in the top left-hand corner there, is we said, how big does a unit outbreak, the same unit, typically get using our retrospective data without interventions? So you say you have 2 patients, we would detect the outbreak on the same unit, usually gets, 2 more patients, 3 more patients, another patient. And we took that data and we modeled it against the outbreaks we saw in the prospective data using the same routes of transmission to say, how big do outbreaks would we expect them to get? And what we did is we said, okay, well, how big do the outbreaks actually get on the same route of transmission, and did a simple expected minus observed calculation in a modeling program. What we found is that in real-time genomic surveillance, we modeled that we prevented 62 infections, which represent 21% of the transmission events, which was what we modeled in our retrospective data that we could have prevented if we intervened in real time. So our retrospective modeling data was aligning with what we modeled in real-time genomic surveillance. Similarly, we also modeled that we could have had we had net cost savings of $700,000 in the 2 years that we did real-time sequencing, which converts to a 3.2-fold return on investment, meaning that for all those amounts of dollars that we spent sequencing, we found net cost savings and a good ROI from our patient infections there. We did some sensitivity analysis to say, what if our outbreaks were smaller or bigger? Running some simulations of, maybe our interventions weren't as good. We still saw 98% of simulations, of 1,000 simulations that we ran, that we would still have net cost savings. And we might have had more net cost savings as well, upwards of 1.7 million US dollars in the same time frame too. So I had mentioned briefly the clusters with no epidemiological links. So our prior threshold was using 15 SNPs, which is a measure of genomic relatedness. We did a pairwise analysis to say, how from were we overcalling our SNPs, meaning were our higher pairwise SNPs mean have no epilinks and our lower pairwise SNPs have epilinks, and that's exactly what we found through this. We found a statistical significance. So from this, we've determined that we actually need to lower our threshold of genetic relatedness, and we now use 10 SNPs. So in summary, from this study, we've been able to show that genomic surveillance detects these outbreaks early that from our prior data most likely would have gone completely undetected. We're able to help direct our infection prevention department to make interventions and observationally stop these outbreaks 95% of the time. We modeled that we prevented multiple infections, possible deaths as well. And we do think that these modeling estimates are conservatives for a lot of reasons that we actually outline a little more in depth in the preprint as well. And we have net cost savings during these past 2 years of preventing these treatment costs of the cost of treating infections versus the lowering costs to sequence a single infection isolate. So I work with a great team here, our EdTech team. Dr. Lee Harrison is the head of our lab, and we have a lot of people that work in our lab and collaborate with us, but also our infection prevention team, who I used to work with a lot of these folks as well, where this could not be possible without all of them, which is I really try to highlight because we can't just sequence and try to reduce infections. We have to sequence, make interventions to reduce the infections, and that's the result of this really great team here. So happy to chat here. If you don't want to chat here, my contact info is there as well, but we've got plenty of time to open it up for questions, this is the part I really like doing as well. So thank you.
Thanks so much, Alex. Really fascinating stuff. and I made lots of notes. It's just really some really nice graphics in there. I loved what you did with the retrospective analysis, where you had all the different pathogens and you had them grouped with the different colors. I thought that really stood out.
Thank you. Yeah,
yeah.
That was my dissertation project, and my wife for my graduation printed it out and framed it for me. So it's in my office right over there because I loved it so much.
What a nice thing. And I think for me, it was just great to see how you had a complete picture there. You know, you'd gone from implementing the technology in a real world setting, and it must have been so satisfying to have those tangible benefits where you could actually go and detect the outbreaks and then all those benefits for patients. I guess my first question before we go to the audience, and John's going to talk about the poll in a minute as well, is this now starting to be implemented routinely in the US? Because a lot of people are talking about the use of these tools for IPC and outbreak detection, but is it actually starting to be embedded in kind of clinical practice?
So I think, and this goes into John's poll there as well, that the evidence base is really there, not only from us, but folks in the UK, Sharon Peacock, folks in Australia like Pat Harris are really showing that this is a good IP practice. We're not nearly close here in the US to being this is a routine practice. To my knowledge, our hospital, and there's a hospital in Miami, are the only hospitals in the US to be doing this. And our hospital is grant funded through the NIH. We don't have any funding internally whatsoever to do this project. So I think those are some of the barriers that we can get into. But I would love this to be routine practice, because we clearly see from this and other studies that our current practices, we miss a lot of outbreaks.
Yeah, great. Thanks, Alex. John?
Yeah, miss a lot and overcall a lot as well. And think about all the precious resource that we're spending on managing outbreaks that aren't even outbreaks at all.
Right.
So, I mean, it's amazing, isn't it? Only 2 hospitals in the whole of the US doing this approach, despite some pretty good evidence. And we'll get on to the discussion about why the barriers for adoption in a minute. But a couple of questions about your study and the methods. One from Kerry, thank you. In the cost saving calculation, do you include the equipment required?
Yes. So to Kerry's response, I'll link here also a preprint, which is under review currently, where our lab folks outline very clearly and detailed the laboratory methods, the bioinformatic methods, everything that we use. And we did a cost analysis in there. And in that cost analysis, we included an FTE laboratory technician. We included part-time FTE of a bioinformatician. We included all the material costs of everything we do each week. And we showed also how we optimized it to reduce the costs. And that cost with FTEs is about still $90 per isolate. And we show in that preprint, which is under review, that for some folks that may not be too familiar, is that if you only sequence a couple of isolates, you're not filling a flow cell and you're actually wasting materials. So if you're optimizing and filling that flow cell with lots of infections every single week, you can reduce your costs really low. We got low as one week, it's been a minute since I've reviewed our paper again, to about like $40 an isolate in a week. So, typically we used to think the cost of whole genome sequencing is like $200, $500 to sequence something. That cost has really come down. And I'm hoping that, through sharing that preprint as well, that we can highlight that, that it's very affordable. Now, we did that mainly in-house. So our research lab, we do library prep in-house. We prepare our libraries, but then we actually send out to a commercial company for the actual sequencing. We bring that data back to our lab where we have a bioinformatician with a very automated pipeline do that analysis, but we don't do everything in-house, nor do we expect anyone to ever do anything in-house. So take a peek at that, and I'm hoping that that'll help some folks maybe explore some options of what could be possible there.
Thanks, Alex. Another question from the audience. Do you sequence isolates from invasive infections only as opposed to patients who are colonized with the respective bacteria?
Yeah, so we do, for the purposes of this paper, it's all what we call clinical infections. So yeah, actual, infections from different bodily sites, blood, sputum, wounds, stool, urine, and things like that. So no colonizations in this study. A typical academic. I'll also link to that comment there, a preprint also under review right now, where we did genomic surveillance of VRE, of colonized patients, as well as our clinical infections. What we found, at least with VRE, is that if you aren't sequencing the colonizations, you're actually missing a big picture. We have a lot of colonized transmission, which some of them, the clinical infections are part of those outbreaks, and it helps us better understand how the infections are spreading. So if you only look at clinical VRE infections and sequencing, you actually might be missing some of that picture. So I had mentioned in our paper that, we had 5 outbreaks that had failed interventions. Well, 3 of those outbreaks were from VRE, which from this paper we know is heavily driven by colonization. And in our interventions, we're not measuring colonization or looking at that as well. So that's why we think those outbreak interventions failed there. And then you might see a similar story with MRSA as well. A lot of folks do MRSA surveillance and sequencing. We haven't quite looked at that yet, but there are some limitations to our paper, of course, that, we aren't looking at the full picture of those colonized with these pathogens.
Okay, great, Alex. Thanks. Let's have a look at the poll results. So the question that we put out was this: rank the following as barriers to implementing proactive whole genome sequencing or genomic surveillance in IPC. So the options were lack of infrastructure, too expensive, lack of expertise, lack of defined and agreed methodology, limited evidence of cost effectiveness, and limited evidence of preventing transmission or infection. Just to qualify, I didn't put cost in there twice. I did mean to put too expensive and limited evidence of cost effectiveness. I mean, clearly cost is balanced against effectiveness to measure cost effectiveness. So it's different angles of the same thing. But I think in the minds of some, it might just be too expensive to even be a starter. So, Alex, what do you think about that ranking? Are you surprised, or is that what you'd expect to see?
Well, my first thought when I looked at this was like, okay, maybe I'm at least doing my job well if the evidence is the lowest ranked responses there.
That's a good point.
But the lack of infrastructure, too expensive, lack of expertise. I think this is where people think, like, how do I even start a program like this at my hospital? Because we're an academic center, we have access to a lot of people and stuff like that. But I think this is where, in the future, when this is more commonplace, I think there's going to be a lot of partnerships between healthcare, public health agencies, and commercial entities as well. Because you don't have to have your own Illumina sequencing machine or nanopore sequencing machine in your own hospital. There are commercial companies or public health agencies, some places, that can allow you to send it to them, that have the infrastructure, that may be able to offer it at a lower cost, and provide analysis that you don't necessarily need that expertise. So I'd be interested to hear from some of the stories or some of the views, if any of the folks online now would want to touch upon why they think their infrastructure or might be too expensive is there. Because from our own experience too, we're able to show that we can get this cost pretty low. But I was talking to John and you and Phil right before this, is that we're still fully NIH funded to do genomic surveillance. Our hospital isn't paying for genomic surveillance. So, I think we have to discuss, how do we convince our hospitals to pay for this approach if we actually want to make it more widespread practice.
Yeah, and we all know that spending to save money is never a popular approach in healthcare. So even if you do have good, strong evidence of cost effectiveness, if there's an upfront cost, it's going to be pushing water uphill with a stick to an extent to actually extract the money from the organization that you need to set things up, especially when there is such upfront infrastructure cost associated with this. Right, Phil, right. What do you think? Were you surprised by this ranking?
I think I was I thought that costs would come up into it, definitely. And like Alex said, it was nice to almost see that the evidence being the most, towards the bottom. So I think it was to be expected, really. But I think there's more and more work coming out, isn't there, that's showing this can really be cost effective and can really make a difference. So hopefully the tide is beginning to change. But I was quite staggered by Alex's fact about only 2 US hospitals, implementing this. And I think, it's probably a similar state of affairs in the UK, where there are very few people who are actually doing this on a routine basis. Do you want to stick with the poll, John, or should we go to next question?
We just one more point to make, and of those two, 50% of those two in the States sound like they're getting it as a freebie as well. So that's another thing to consider.
Yeah.
So this ranking is really interesting. I actually think this is exactly the same ranking that I put in. So it's interesting to see. And lack of infrastructure and cost and expertise. I think many people working in the community have a bit of a blind spot for sequencing in general. It's just a language that they don't understand. They're not used to the terminology. They don't know how to interpret the data when it's there. So I think there's quite a bit of anxiety is probably the wrong word, but it's the best one I can come up with about whole genome sequencing within the IP community, particularly because it's such an unknown and because it's not many people working in infection prevention haven't come directly from a laboratory background. So I think there's a big education piece for Alex and colleagues just to get the messaging out there that actually when you've got the right infrastructure and the right personnel in place, it's not hard, it's not complicated, it's straightforward, and actually it's really impactful.
Very much so. I mean, I agree fully. And we talk about education. This is something I'm trying to work on with APIC here in the US, is that, there is no training or even modules for IPs here about, how to use sequencing or review it. And I think at the end, we're not asking IPs to, run the bioinformatic analyses and look at a SNP matrix or anything like that. What I send to our infection prevention department is, here are your patients in an outbreak. That's it. And I think whenever we communicate that with IPs, that's what we want to see. We don't necessarily need to see gene presence, absence, and all these other things, which is really interesting from a research perspective. But more so just binary, here's your outbreak, here are your patients, let's go investigate that.
Yeah, I think that's a really powerful way of simplifying, the outputs that you get. And I think that's really how IP can make decisions based on information like that. And I agree exactly with what John was saying. Some of the terminology, it can feel a little bit scary for people who aren't in that world. But actually, when you show that you can set it up effectively, it can really pay dividends, can't it? We've got a lot of questions coming in, Alex, so I'm going to go back to those.
All right.
Damien says, thanks for the great talk. How did the infection prevention and control team feel about the results showing difference between suspected and WGS confirmed outbreaks?
I mean, so they often get surprised because for what they thought were an outbreak of like, oh, we have like 5 MRSA patients on this unit, this is definitely going to be an outbreak, or like stenotrophomonas on this ICU, this is going to be an outbreak. And so many times we've just showed them, they're 1,000 SNPs apart, they're not related. So oftentimes they're a little shocked at those results. And then when we show them that, these are the outbreaks that you're missing, but we're also able to identify sometimes very clear links between these patients, in a way makes them feel better because, we used to respond to these suspected outbreaks, put all that energy and effort and realize that, it wasn't really all worth for it. But now they know, they have confidence that when they're intervening on an outbreak, we know it's an outbreak, and typically they feel better that, they're spending that time and effort on there. And speaking of time and effort briefly, we always say, look, isn't this making so much more work for your IP department? But to that same point is that we used to chase our tails of what we thought were outbreaks and waste resources. Now we know that the resources we're spending on outbreaks are true outbreaks. And we are able to show now too that we're making an effect at reducing infections. So what we're doing is both worth it, even though we're spending the time on it.
Yeah, again, just before John comes in, I think that's a really great way to look at it in terms of changing the dynamic a little bit, and you think those outbreaks that you were going after weren't actually outbreaks, and the amount of time and that goes into those interventions, the education, the time and resource for the IP team. So I think that's a really good way to look at it as the other advantage that it has. Over to you, John.
Yeah, I'm not surprised that this gets a warm reception from IP teams. I mean, we're not stupid, are we? We know there's a lot of uncertainty around calling outbreaks and calling transmission routes. I was involved in an outbreak today, and we just didn't know whether the outbreak was from this bay here or from a community exposure here. And having that resolution of whole genome sequencing would certainly take away some of that uncertainty, and I think help us to be even more credible, actually, with our clinical colleagues, rather than all this uncertainty and maybe over intervening in the context of that uncertainty. Right, next question. Great talk. I wanted to ask if in your hospital, do you do any routine environmental sampling? And do you think that this could be of added value in order to identify outbreaks early?
So currently we do not. Although in the past month, I've just gotten 2 grants to start doing environmental sampling with the sequencing to add it to our data. So very excited to move that forward. But I think, at least from my own anecdotal, views of some of the literature, I feel like here in the US, environmental sampling isn't, done much at all whatsoever. And we do have to ask the question is, what does it add, especially in the lack of sequencing? If you have an outbreak that's already occurred and you have a positive sample from, a bed rail, did that patient put it on the bed rail, or did the bed rail give it to the patient? And then without sequencing, is it even the same species? So I do think there could be a value, which is hopefully why I got some of these grants, to combine prospective environmental sampling with sequencing of clinical infections, because we can look downstream of after we have positive samples, repeat positives of, okay, we have a positive environmental sample that is matching a future clinical infection. Now we can show directionality. And there's been some great papers out of the UK that have shown that. And I'm especially interested in water. Mark Garvey, I love a lot of his papers out of there in the UK, has really got me excited about sinks and splash zones and things like that. So one of the grants, a smaller grant that I just started, is culturing water sources to look for Pseudomonas and NTM to match it to some of our sequencing. And I'm excited to see what that finds, because I think there's a lot of great preliminary data out there that shows we're giving it 2 patients from water, which has been built up by some of those UK papers. So I think there is a use for it, but I don't think it's clearly defined yet. And that's what I hope some of the literature can show soon.
Great, thanks, Alex. Yeah, Mark was a previous guest on IPC Partners Journal Club, and he talked about the splash zone, which is just a really wonderful piece of research. Quite simple, but yeah, I'm really excited to see if there's links to, looking at outbreaks and tracking transmission with this technique around water. It's a huge challenge in the UK. Okay, back to the audience. Sally asks, could this be valuable for distinguishing bacteremias, UTIs, infections, etc., I guess, that are endogenous from those that are due to breakdown in IP practices? What do you think?
Yeah, I think so. I mean, I think when we think about those that are colonized and then you get your infection, and there's some good data published, I saw in CID, where especially for CRE, CPE, CPOs, that people have shown that, you're colonized with that bacteria, you sequence it, and then those patients at X time frame become infected with it. I think that's a very useful tool to show clinical progression, and especially, where are these patients getting it from? Are they getting it from other patients, or is it just their own, like Sally had mentioned, their own endogenous bacteria, where we're doing something to them which is causing their infection? And there's clear use for that, because that's IP practice to prevent endogenous flora from going into patient, central line care, Foley care, things like that. Whereas what my research is mainly trying to stop is through environmental transmission, and that's a different intervention. So there's definitely a good case use for looking at that data as well.
Yeah, I think there's sort of an advantage, isn't there, where you have isolates from everywhere, every conceivable potential source. You've got the environment, you've got patient skin, you've got patient body sites all over the place, you've got visitors, and then you have complete resolution. I think the reality is that's not going to be feasible for an ongoing prospective surveillance project. We need to probably select key isolates, key organisms, key presentations in the of the organisms. Otherwise, things are going to get out of hand in terms of cost. Okay, back to the audience. So Barley is asking a question that was on my mind too around SNPs and where we draw the line and how we call it. So the number of SNPs required has varied depending on the context, and you mentioned that you actually varied your SNP threshold in response to the findings to make it make sense. Are there now standard guidelines and an international consensus on the number of SNPs needed to determine relatedness?
Oh, that's a $1 million question, Barley. You know, I previously did a systematic review on genomic surveillance studies and looked at SNP thresholds and just found wide varieties of what people called SNP thresholds. You know, some people, there's one study I believe that used 90 SNPs to call relatedness for Staph aureus, which is so high. And then, you know, C. diff, which is a different bug, we go down to like 0, 1, 2 SNPs. So I definitely think that there is a, you know, organism specific potential there as well. You know, we try to show that, and what we have always said is that you have to both look at SNPs and the epidemiological context as well. Because oftentimes we still find 0 SNP clusters with nothing in common between them. And there you might be missing some intermediate isolates as well. So I don't think that we're ever going to get to, you know, 10 SNPs is genetically related. I think we always have to look at the big picture of what's the epi context in there as well. And then I think too, when we talk about, you know, bioinformatic pipelines, which is how we analyze these genomes, you know, we mentioned this in our commentary that we just put out in ASM that I mentioned in the blog post, is that, you know, I don't think the point is to necessarily everyone uses the exact same pipeline, threshold, etc. I think the purpose of going forward is that can we all agree that we might come to essentially the same or very similar conclusions if we were to do it of who's in our outbreak, who's not in our outbreak. And I think that's going to be the downhill or the uphill battle that we'll have to push for to get things more systematic in a sense.
Thanks, Alex. One more question, which is from me, and hopefully this will just bring us to a nice close. I remember when we first started sequencing bacterial genomes and the price that was involved and the time and the resource, etc. Given, you know, how far we've come in the last, I think 30 years since the first one was Haemophilus influenza, I think 95. Given how far we've come, and, you know, you talked a little bit about the speed and the price, for someone who's at, you know, the cutting edge of this technology and talking about implementing it within healthcare systems, where do you think this technology goes next? Does it continue to get faster, cheaper, or are we going in a different direction?
I hope faster and cheaper for both reasons. But I think for adoption, at least here in the US, the battle that, you know, we've realized that we've kind of shifted our fight to is approaching healthcare payers, insurance companies here in the US, private, you know, public insurance, because we you know, kept trying to convince hospitals, you're going to save all this money, you're going to find these infections, make your patients safer. But we thought about who's saving the money, and it's costs saved from the cost of treating infections. And, you know, hospitals don't realize those savings. It's the payers who are paying for that treatment. Especially here in the US, those are our insurance companies. So what we've tried to shift to is approach these insurance companies, approach, you know, these payers and say, if you can incentivize your hospitals that you're using, their patients will be safer, and you will see that 3.2 return on investment if you can get your hospitals to do this, because we haven't been successful at convincing hospitals to adopt this, mainly because, you know, they're not seeing that. You know, how is this going to become widespread? There's a separate pathway here that we didn't touch on at all, of using genomic sequencing for AST prediction, which there's some preliminary data that might be more accurate than what we do right now. You know, there's a lot of regulatory and operational barriers for that. But if sequencing for infection prevention takes off, that's going to benefit AST prediction, or if AST prediction takes off, that's going to benefit infection prevention. So, I think there's going to be a tipping point. I don't know exactly what it is. If I have a crystal ball, that would be great. But I feel like in the next 10 years, we're going to hopefully see it take off here.
Great. Thanks very much, Alex.
Yeah, thank you, Phil. Thank you, John.
Okay. Thanks everybody for joining today. Thank you especially to Alex for joining us. I thoroughly enjoyed that. I think the Q&A was fantastic. So thank you everybody who was able to join us and to submit questions. Next up, we have got a Journal Club special edition in partnership with HIS in a couple of weeks' time, where we'll be picking up some of the themes around water that we touched on in this Journal Club today. So it's in the water, addressing risks from healthcare water systems, and upcoming Journal Clubs on health inequalities with Kerry Holden, natural ventilation with Alex Edwards, and a bit of an update on Canada Aris from me later on in the year. The schedule for the future Journal Clubs is kept up to date on our website. All recordings are available from YouTube from these Journal Clubs, and you can thank you to IPC Partners for hosting the event. So thanks again. We'll see you all soon, and Happy New Year. Bye.
Thanks, John. Thanks, Alex.